nice introduction. So I started sharing my screen. I guess you can see it. So, and also thank you for the invitation to give this talk to the network. So today I will discuss a little bit about the computational methodologies that are used for the discovery of cryptic allosteric pockets and the latest progress from my group. So let's start with a general introduction. First of all, a motivation about why we are interested in cryptic pockets in general, even if this is not needed for most of our network participants. So basically, we know that the human genome contains about 20,000 protein coding genes, and a fraction of these are important disease modifying genes like oncogenes. However, only fraction of these disease modifying genes are druggable, and druggable we define it as two things: ligandable, so we can bind to them with a drug-like molecule or a biologic, and then modifiable, so we can modify their activity after we're bound to them. So because of this, we are in dire need of approaches, alternative approaches to try and target these important disease-modifying genes. One of them, it's targeting cryptic pockets. So cryptic pocket here show a clear example. This is an enzyme where you see that at this level between these two helices, there is no cavity. However, crystal structures have shown in a completely unexpected way, serendipitously, that you can bind actually two small ligands at this interface. So this is a typical cryptic binding site. And their definition is a bit fuzzy. So typically they are defined as binding sites that are cryptic, so from Greek hidden, so not visible in the protein targets crystallized without a ligand, and only become visible crystallographically or anyway by NMR or cryo-EM upon a binding event. So most of this cryptic pocket sort of open like this one. So there is an opening of a cavity, the creation of a cavity when the ligand binds. But there are some cases where the cryptic pocket actually closes around the ligand. And so already this creates a difficulty for many approaches, including computational approaches, because maybe an approach that works for opening a cavity does not necessarily work for closing a cavity around the ligand. They can, of course, provide alternative to substrate competitive pockets, and there is the case of RAS, a very important oncogene that I will discuss in a minute, which was thought to be completely undruggable until recent times. And also, they seem to be more common than previously thought. So methods using machine learning, for instance, predict that a large fraction of the human proteome might contain cryptic pockets. However, much still is not known about them. Most of them have been found serendipitously, so by chance, and it's not trivial to design ligands in a rational way for them. How many examples are there in protein data bank? So one of the first repositories that then use this information to design a machine learning approach to find them was CryptoCite, that reported about 90 examples. So it's a very small fraction of the proteins deposited in the protein data bank. Then a study by Beglow et al. From the group of VIDA found that actually of this 90 example, 45 are not really cryptic in the sense that the cavity is visible in at least one of the deposited X-ray structures. So, but how they were found, and are there other examples in the protein data bank? So you might imagine that, well, we could just take the protein data bank and select all the ligand-bound structures and then try to see if the ligand opens cryptic pockets or not. But this is not trivial because, first of all, what do you analyze? You can analyze the pocket opening, volume expansion, or decreased pocket enclosure, or if there is a clash with the ligand. However, there are a number of problems because for many structures in the protein data bank, we are missing electron density, especially in the flexible sites. And typically, these flexible sites are those we are interested for the cryptic sites. Then there are some conformational changes that are too large. So if you operatively try to align the protein in the apo and the hollow states, this alignment it's error prone. Then you have many cases of protein-protein interaction, and these are particularly interesting, but you have to consider each individual chain for the alignment, and you will have alignment issues. Moreover, because now we are trying to do this ourselves in our group, there are many problems with non-standard naming of residues. How do you consider, I mean, ligands? Do you consider everything that is not a protein as a ligand, or you do not consider small molecules that have added for the crystallization of ligand, etc. So because of this, there is a paucity of data. So actually there are these 90 examples and a few more. So I would say less probably than 200 examples are well known. And this clearly complicates the use of machine learning to predict where these cryptic sites are. So as I said, but this is a work in progress, and maybe I will talk about this next time because still not finished. We're trying to reanalyze all the protein data bank and clusterize all the structure there in protein families according to their sequence similarity. And we see that by using a robust scoring function, so that we see the clash with the ligand, not just in a protein, but only in the deposited protein. So the cryptic pocket is one that is only visible when the ligand is bound, that there are more than 250 cluster of proteins that harbor, there is a strong signal that they harbor cryptic pockets. So there are maybe more than those that we directly know through cryptosite and other existing sites. But still, this is work in progress and is less trivial than you might think. So an important question is, do the cryptic pockets have a functional role? And there are some reports in the literature, for instance, this report from Novarek et al. 2021, where they've seen that a cryptic pocket opens under the omega loop of beta-lactamases, and the results from this suggest that excited state containing cryptic pockets can be advantageous for function. So actually, these are functionally important. And this also has a consequence for drug discovery, because if they are functionally important, then targeting these cryptic pockets is more interesting for drug discovery. Because of course, if you target a cryptic pocket where you can bind a small molecule, but there is no modification of the function of your target, in this case, it's a cryptic pocket that is not interesting for drug discovery. But if they are functional, on the contrary, obviously there is an interest in targeting them to enhance or suppress the function of your target. That is the question of cryptic pockets of protein-protein interfaces. So we know that most of the proteins in our cell engage in protein-protein interactions, and they're very important for the cell homeostasis and to regulate all the cellular function. And because clearly the number of this protein-protein interaction, it's huge when you consider all the possible interaction between the 20,000 or so human coding protein genes, you see that there are many, many opportunities for drug discovery. So one of the questions is, are protein-protein interaction prone to the formation of these cryptic pockets? And there are some hints that this might be the case, because we know, for instance, then when protein-protein interaction is formed, flexibility is often an important plays an important role, because it's not just the rigid docking of two rigid proteins, but there are actually changes at the surface that can be small, like the rotation of lateral chains, but can also be much more important. So if that's the case, if there is flexibility between the two proteins, we can imagine that this opens the opportunity for cryptic cavities, like shown here. So this is the interleukin-2 in gray with the interleukin-2 receptor, and basically there is in green a possibility of binding a ligand to a cryptic pockets at the interface. But what is the problem? The problem is that if we use most of our pocket prediction computational machineries that we have, they predict actually the cavity on the other side of this interaction. So they are not very useful. So designing drugs that disrupt its protein-protein interfaces, it's particularly challenging, because they are often flat and featureless in the apostructure. As I said, conformational changes are all often involved. So these transit or cryptic pockets play a role in most cases, but it's not trivial to predict where they are, and sometimes they are maybe too superficial to design potent ligands. So from an experimental point of view, one of the best approaches that we have, it's tethering. So if we can covalently bind some ligand with a more or less flexible linker, then because we highly increase the local concentration of the ligand, because now it's really bound to your protein, in this way you can screen against a library of different ligands for the presence of cryptic pockets. Clearly, also fragment screening with various approaches, from NMR to also soaking with crystallography, are also good approaches to find cryptic pockets in principle. But there are problems there, because as I will show you in the case of a target for SARS-CoV-19, if you have a pre-crystallized structure, sometimes the crystal packing blocks the opening of the cavities, of the cryptic cavities. So I wanted to show you this famous example of a success story with the cryptic pockets, and it's the one of RAS. RAS, we all know, it's one of the most important oncogenes. It's in many solid tumor, and typically when you have mutations of RAS, the tumor evolves towards more malignant phenotypes. So this is RAS bound to the physiological ligand, which binds very tightly to the cavity. So for over 30 years, because of this very tight binding, it was thought that KRAS was undruggable. But then, thanks to the work of Shakat and others, they found the first covalent inhibitors of KRAS mutant J12C, whereby covalently binding to this cysteine, they were able to find this cryptic cavity. And now there are a number of drugs, either in clinical trials or already approved, that use this pocket to bind to KRAS. So, because experimental approaches are complicated, as I have shown, and not always successful, because especially if you do not have a cysteine or another residue close to where the cryptic pocket is airborne, it becomes very difficult to covalently target the surface and in this way find cryptic pockets. And for the problem that I've told you about soaking crystallized structure, etc. So computational tools in this case can play a very useful role. So for instance, here there is a movie, if it works, yes, of a molecular dynamic simulation of a ligand crawling to the surface of interleukin 2. This has been, this is a paper by this show research group by Bing Shan and others. And actually, by crawling on the surface, after very long simulation, you find that the ligand is able to find its bound state that is very close to the crystal structure. So you see, if we can do this, this is very, very helpful for finding cryptic pockets. So of course, molecular simulations, I do not think that they need an introduction to this audience, but just quickly to say that they are based on Newton, to resolving Newton equations with an approximated functional, a force field shown here. And this force field, basically, they are all date back, they are all in a way or another descending from the Lifson type force field that has been developed almost 50 years ago. It's a very simplified force field where you have springs and you have torsional angles and you have point charges. So it's quite approximated. But for how approximated it is, it really does a good job to, let's say, approximate in a mean field way all the important contacts. And so they are surprisingly accurate, considering how simple this functional formula is. So once you have the simulation of your, for instance, hypostructure, you can use algorithm to detect transient pockets. And there are a number like epoch, F-pocket, trap, POVME, nanoshaper, etc. One problem is that if you run a long simulation of your targets without ligands, these transient cavities do not open up. So even if you run, nowadays you can do tens of microsecond long simulation, you don't see the opening of the cavity. And this is because in many cases there is an important component of induced fit in the opening of the cavity. Of course, the mechanism of opening of this cavity, whether it's an induced fit or conformational selection, is still an ongoing discussion. And it could be different for different cases. But we have several hints also from simulations that actually there is an important component of induced fit. And this is why when you run your simulation with mixed solvents, like water with isopropyl alcohol or water and benzene, resorcinol, acetic acid, and there is a large literature about mixed solvent simulations, often these are much more effective than just running with water your protein. So you have this mixed solvent and then all these small fragments that can be benzene or isopropyl alcohol or this sort of small molecules. They encourage or stabilize the opening of these cryptic pockets, so they are much more easy to find. One important question that we have to ask in the community of computational chemistry, it's whether the force field it's good enough for cryptic pockets. And this is not a trivial question. So I have briefly explained to you that force fields are approximated, and the functional form that we used is the same, has been for the same for 50 years, not for lack of trying, but just because it's sufficiently accurate for most cases, and it's very fast to run. Whether more complicated force field, like with polarizability, they tend to be much slower. But then the question arises, so this force field have been parameterized to try to capture the properties of well-folded proteins, like here in a scale, proteins like ubiquitin or GP3, that if you look by NMR, they are like rocks. They're very rigid. But this force field, we know that work much less well for intrinsically unfolded or partially unfolded proteins or domains like alpha-synuclein. Because we want to observe the opening of these cavities and the changes, for instance, of very flexible loops. So a question that we might ask is the force field good enough for the opening of these cavities, or it predicts too stable structures? And in fact, this was a problem with a force field as Amber 99 SB or Charm 36. Now, luckily, many papers have reported that more recent force field like Charm 36M, this Amber, Amber DISP, or latest version of Amber are more balanced. So they are able to capture well both the well-folded and also intrinsically unfolded parts. For instance, many people looked at this. We also looked at this in combination with NMR for the full Winterbrand factor, for instance. These two domains important biomolecular target as one part that is very well folded, and another part that is like a molten globule here, where you have disulfide bridges keeping it together. And if you use a force field like Charm 36M, that is being reparametrized to also capture intrinsically unfolded, partially unfolded domain, you see that when you look at the NOE violation with NMR, picture is quite good. So I would say, when you look at the opening of cryptic pocket, I urge you that you use force field that work well also to capture partially unfolded domains, or anyway, the flexibility, not just a very rigid domain. So say that the problem that we have with simulations in general, but also with cryptic pockets in particular, it's the time scale problem. So in the simulation, we have to integrate the equation of motion for numerical stability in the femtosecond regime, while often the opening of the cavities happens on much longer time scale. So some of these cavities, like you have the one you have seen for interleukin, it's rather superficial. You have the reorientation of some lateral chain. So few microseconds are sufficient. But in other cases, we have cases where you really have to change secondary structure, like the unfolding of alpha helix or beta sheet, or very large movement of loops. In this case, the time scale is longer. And so brute force molecular simulations, even when helped by a mixed solvent, are not sufficient. So this can be seen in this scheme here. So we have the free energy, and your protein starts in a well-folded conformation. If then you want to reach a conformation that it's partially unfolded, or where there is a cavity, you have to overcome a high a high barrier to the transition state. And if this barrier is sufficiently high, this will never happen in your simulation, but also in the reality, because the probability of being on the transition state depends on the exponential of the difference of free energy between the bound state, the folded state, and this transition state. So because of this, many methods have been developed in molecular simulations. There are specialized supercomputers like Anton 3 that amazingly can push 100 microseconds per day on a million atoms. And there are methods that can take can run on GPUs and more normal architectures based on multiple replicas with temperature, like Hamiltonian, like parallel tempering, or with multiple Hamiltonian, like Hamiltonian replica exchange, the method that we developed, Swish, that I will talk about in a minute. Or you can use adaptive sampling, like the Markov state models that I use much in the Bowman group that also looking at cryptic pockets, or also in Gianni De Fabritis group. And of course, you have collective variable-based methods like umbrella sampling, metadynamics. The problem there is that you need to have a good collective variable to describe this. One possibility is JEDI, developed by the group of Julien Michel, but these methods are still a lot under development. So what is the way this method works? For instance, if you have a collective variable-based method, like umbrella sampling metadynamics, you can fill the free energy landscape as a function of this collective variable until you explore all your states, including the open cavity states. So I will show you some of our progress in trying to understand the nature of the cryptic pocket opening with molecular dynamics, testing the predictive value of various approaches, including log molecular dynamic simulation, and then what was our thinking around designing general algorithms to find cryptic pockets with simulations. So we initially started looking at this problem several years ago, now 9 years ago, together with my, at the time, PhD student, Vladas Olenikovas, and in collaboration with the UCB. And Benjamin Kossin, at the time, he was at UCB, and he really started the discussion with us. So we took these 3 cases, the beta-lactamase 10-1, the one I showed you before, that is important for drug resistance. Then interleukin-2, the other case I've shown you before, it's important for the regulation of the immune system. And then another case, here, it's NPC2, and we also look at P38 alpha, etc. So the first, this is already nine years ago. First thing we looked, we looked what happened with just running long molecular dynamic simulation of the APO protein with different force fields. So we ran, in this case, 1 microsecond long simulation. At the time it was pretty long, now it's much more standard. And then from this, we ran other 5 simulations. So what is this metric here? This is pocket exposure. So basically, we take the pocket that we know from crystal structure, and by running from the hollow structure, we see in how many cases we reach at least 80% solvent exposure of this cavity. So if we go above this 80% threshold, we say that the cavity is open. And so you see that in all this long simulation, maybe there was one instance where for a very short time, there was this opening of the cavity. So I would say that this is not very successful. So with different force field, we see that the cavity does not open much. Well, from time to time it opens, and also if you define broadly opening of the cavity, yes, sometimes if you, for instance, say 60% is sufficient, yes, sometimes you visit it. It's not really clear. And then if you didn't know where the cavity was, this wouldn't be very useful for drug design, because you wouldn't recognize a clear cavity there. So if we look at different methods to analyze the pockets, you see that there are some instances of opening there, but also the main pocket, it's predicted to be somewhere else. Not very useful. So in the following, I will use violin plots. So violin plots are similar to box plot, but also you show here on the y-axis the probability density. So they're very useful because you don't only have the statistical values, but also you see the profile of the probability density. So we repeated this with different mixed solvent, with benzene, with heteroaromatic fragments. And we see that if you run many simulation, 100 nanosecond long simulation, in a number of instances, you see the opening of the cavity. But it's not very clear. So you have to really run this for 1 microsecond. And then for 1 microsecond, if you look at all this independent simulation, you have a number of cases shown here, where after 1 microsecond, there is a clear opening of the cavity. But this is clearly very expensive from a computational point of view. So I would say this approach works, but can be too computationally expensive to deploy across many targets with many different fragments. So at the time, encouraged by this idea that basically there is, you have to open hydrophobic cavities to let the fragments of the mixed solvent bind, we thought, well, maybe we can design an Hamiltonian replica exchange that my at the time student Vladas called Swish, whereby you change the Hamiltonian of the interaction of your ligands with hydrophobic of your hydrophobic cavity with the water. So basically, you make the water in a way more hydrophobic, so interacts more strongly with the hydrophobic residues. So the opening of these hydrophobic cavities, it's less penalized. When we did this, and now we run again on the TEM1 beta-lactamase, we see that basically for TEM1, clearly using Swish encourages the opening of the cavity at a high lambda, where there is more interaction with the water, this changed water. But also it's interesting to see that, well, this not always works so well. For instance, for PLK does not work very much. There is some encouragement of the opening of the cavity, but it's not great. Why? Because when this re-exchanges with the lower replicas lambda, the cavity immediately collapses. So what really worked here is that if you combine Swish with small fragments like benzene or heteroaromatic, so what happens is that at high lambda, there is Swish encourages the opening of the cavity, and then once it's open, you can bind there multiple benzene, like in this case, or multiple heteroaromatic molecules, and they stabilize it. So once it's stable, there is this exchange with replicas with more normal water, but still the benzene stays there. So the combination of the two worked really well for these cases. So we looked at more complex system, for instance, NPC2, P38 alpha, and you see here basically you have to move a long loop or basically this alpha helices. So these are conformational changes that are more complicated. So the Niemann-Pick type 2 receptor, it's responsible for cholesterol transport, for instance. So you can imagine that the internal cavity, it's very hydrophobic. So it's difficult to open it in absence of some ligands that stabilizes it. But if you combine the ligand with Swish, we were able to really sample it well. P38 alpha MAP kinase is very important for cancer treatment, as is involved in cancer and inflammatory diseases, and can be allosterically activated by lipids binding far from the substrate site. Again, the index cavity requires substantial backbone rearrangement. The other two systems that we have handpicked to look at this were the LFRR, which is a transcription factor that regulates expression of multi-drug efflux pump, and human phenylethanolamine N-methyltransferase that is important for adrenaline synthesis. In all these cases, here our violin plot. So the first one, it's plain MD. Again, this is the pocket volume ratio. These are many replicas with mixed solvent. This is Swish alone, and this is Swish with probes. So you see that in most cases, just MD does not encourage the opening of the cavity. Maybe P38 alpha, you have something, but not much. When you have mixed solvent, in P38 alpha, you have some successful cases, and also in L and this transcription factor, but not in the other two cases, and especially not in the Niemann-Pick target. Swish works really well with P38 alpha and also in this other target. The combination of the two works well for all of them. So one question that is often asked is, how can you, so you see the opening of these cavities, how can you distinguish real cryptic pockets from false positives? This is a difficult difficult question, and we tried to address it, for instance, looking at the difference between the cavities that were obtained with Swish and those that were obtained in molecular dynamics. So if you subtract the cavity, for instance, for 10 beta-lactamase, the cavities that open in the presence of Swish and fragments from those that you can obtain just by running long MD, in this case, you clearly see where the real pocket is. In this case, it works. In other cases, it might be less effective. So you still need a way to analyze the pockets that you find and to see if they are relevant. So we use this approach during the COVID emergency to explore cryptic pockets in track targets that are linked, that were important for COVID-19. So in particular, we looked at this target that was being less studied. It's a special target. It's non-structural protein 1. It's 20 kilodalton protein, which is a major virulence and pathogenicity factor, as it binds to the 40 subunit of the ribosome to inactivate the translation of function facilitating the host immune response. So it's like a cork that blocks the ribosome. So, and interestingly, it recognizes viral RNA. So it unblocks the expression when you have viral RNA and blocks the expression when you want to have an immune system response. So this is a very peculiar target, very interesting target, but also a very small molecule that is almost featureless. So if you look at it, there are not real cavities. This is something that I have done with my, at the time, student Alberto Borsato, that now is a postdoc in my group. So here is how the non-structural protein 1 works by blocking the ribosome and then recognizing on this N-terminal part viral RNA. So it unblocks the ribosome while the viral RNA is close. So how do we target this? This is almost featureless small ball protein with no binding pockets. So we tried to look with Swish and benzene if we could facilitate the opening of a cavity. And indeed, using Swish plus fragments like benzene, we could see the opening of this cavity. And basically, once we had these pockets located by this approach, we first ran virtual screening campaign to see if we could select some fragments binding to these pockets. And then with X-ray crystallography, in particular with soaking, we validated the pocket together in collaboration with the group of Frank Kosielski at the UCL School of Pharmacy. And this was successful because we could find several of these computational predicted fragments that bind to the cryptic pockets. We could validate only one of them, not the other two. We believe that this is because we used soaking, and because of the crystal packing, in particular this yellow cavity, it's not accessible in the crystal. So we think this is a limitation of the experimental setup, but it's still something that we are looking at. The interesting thing of these cavities is that if you look at the conservation of the residues around the cavities for different families of coronaviruses, these are conserved. So in our opinion, this target is interesting also for future coronavirus pandemic. Fingers crossed this will not happen in our lifetime, but we will always have to be alert. So the next topic is, can we use these approaches to explore cryptic pockets at protein-protein interfaces? So we started looking at these protein-protein interfaces with this simple approach that worked for many globular proteins. So we prepared the receptor in water, so one of the two proteins engaging in a interaction. Then we ran the unbiased molecular dynamic simulation. When I say we, especially I'm talking about Alberto that did all this simulation together with Eleonora Gianquinto that visited my group. Then we ran this with swish with mixed solvent like benzene. And we tried to look at pocket detection. And I can tell you that this was a bit hit and miss. So for some protein-protein targets, it works well, but like interleukin, but for others, it's less successful. So albeit swish worked in some cases, in others, it failed to reveal the pockets. So can we improve on swish? A possibility, so swish is an Hamiltonian replica exchange-based method where we have different replicas. And these different replicas differ by the way water interacts with hydrophobic residues. So can we also add to this multiple replicas temperature? And why we want to do this? Because for instance, if you want to sample the opening of cavities that depends on changes in secondary structure, so a very complicated, something that we are trying to do with Martin in collaboration with Janssen. I cannot still reveal the details, but soon there will be hopefully a paper about that. So swish alone is not sufficient because you have to overcome very high barriers. But if you use temperature, for instance, this is the very famous Diala in free energy landscape. If you go at 1000 Kelvin, you see that the different minima now are joined because all the fluctuation makes it much easier to go from one minimum to the other. So the idea is, can we combine swish with temperature to have the combination of the two opening the cavities? And this is what Alberto with Eleonora and Valerio in my group did. So here we have multiple replicas using with swish, but also different temperatures. Instead of just using parallel tempering, here we used a more refined approach, which is OPES multithermal, where at higher, let's say, replicas, higher lambda replicas, you expand the fluctuations. So you don't just have different temperatures, but you flatten the potential energy surface in a way that you explore multiple temperatures at the same time. So this is the setup. We have holo-like structures and apo-like structures. And for all of them, we run the unbiased simulations, then mixed solvent simulation, normal swish, and finally swish X, the new one with the temperature, in presence of fragments. So to analyze all the cluster of different conformations and different pockets that can open, we used first, principal component analysis, and we took the first 50 vectors out of it, and then we use t-distributed stochastic neighbor embedding to look at how these different minima were distributed, and density-based clustering to cluster all of them. So in this way, we can see the different cluster, how they emerge from different methods. All this, it's now published in JCTC in 2024, if you're interested. So we revisited 10-1 beta-lactamase. Now, with much shorter simulation, now this is only 250 nanoseconds per 6 replicas, or 3 times 500 nanoseconds for the unbiased and mixed solvent simulation. For 10-1 beta-lactamase, we see that basically swish X is the one that works best. if you start from the hollow, of course, initially the cavity is opening, but then it collapses. So this is really just because the simulation is short. If you run it longer and longer, it will stabilize around the closed state. Mixed solvents are also working relatively well. So this is to confirm that Swiss X for these globular proteins works better than Swiss. Now, if we look at the cluster with this TSNE analysis and density cluster, so here more red clusters are the one with the pocket it's more exposed. The contour plot is the one that is obtained from the hollow-like structure that is slowly collapsing. And so what you have to see here, that with C-Shex, you have many more clusters and that are red. So there are many more open-like clusters. In particular, this 22 cluster, if you look at it, really agrees with the crystal structure of the bound state. Swish, there is some opening. In mixed solvent, there is some opening, but not as effective as Swish X. Now, if we look more at the details, this time we color the cluster as a function of the root mean square deviation from the crystal structure. You see that there are clusters that are very close to the crystal structure. So their root mean square deviation is very close to the one of the crystal structure. Okay, so let's look at protein-protein interactions. So as I said before, protein-protein interaction are very, very promising targets, these interfaces. So if you look, for instance, at the mutation that are cancer, that are oncogenic, and you look those at the interface in red against those that are not at the interface, you see that there is a clear enrichment of oncogenic mutations at the protein-protein interfaces. Still, this is very difficult. So the number of drugs that are approved or in development for protein-protein interfaces are few. So can we use leverage molecular simulation-based tools to predict good cryptic pockets at protein-protein interfaces? So we looked at this target, BCL-XL, that is important for apoptosis, interleukin-2, MDM2, and HPV-11A2. And basically, because this isn't much time, I will rush a little bit. So the only effective approach for many of these targets is Swiss X. So if you if you think that this unbiased hollow-like, because it's short, initially it's open, but then it collapses. So the only one that really works is Swiss X. So the cavity is really well exposed. And if we look at now again the cluster colored with pocket exposure, even Swish, you have very few cases where you have some partial opening of the cavity. Swiss X, thanks to the help with the temperature, works really well. So this is what the predicted structure with Swiss X, this cluster 11 is, and this is superimposed to the crystal structure of the ligand-bound state. So you see that it really works well, the cavity opens well, and you can really do docking and virtual screening with this cavity. This works well also for the other cases explored, like interleukin-2, MP2, and HP11E2. So our general conclusion is that this Swiss X, that is a further development of our Swiss and Hamiltonian replica exchange approach, especially when we combined with fragments, offers a significant speed up, because first of all, the simulations are much shorter than the one that we run with Swiss, but also is very effective in cases like complicated protein-protein interfaces where other methods fail. So now I would like to acknowledge the sources of funding, especially Swiss National Science Foundation. We have a project for this. but also I would like to thank ALLODD for giving me the opportunity to interact and collaborate with many different full members of the consortium, and in particular Zoe for running the consortium so effectively. And this is a picture from a retreat in the mountains in my group, and particularly Alberto, it's there, that ran most of the simulation, with Eleonora here. And then also my collaborators, Josep Aiter for the COVID target, and Ben Cossin that started all this with us. And thank you very much for your attention.
Thank you very much for a very interesting presentation. I think now it's time for some questions that I'm sure a lot of the audience has. I think I could start with the first one. In the beginning of your presentation, you mentioned that you're working currently in your group on PDB analysis to identify the protein clusters that might have cryptic pockets. So I wanted to ask if, from your experience, are there any structural motifs or perhaps protein families that are more prone to having the cryptic pockets? Because you said that they are conserved, for example, for virus families that you studied.
Yeah, so this is an analysis that was an interesting analysis on this has already been made by a group of Sandro Vaida, but also the cryptosite people. So they have seen that there are different classes. Some of the cryptic pockets, like the interleukin 1, are due to the moving around of lateral, so the amino acids. Basically, they open the cavity, superficial. But there are also moving of loops, like I've shown some cases. There are a number of cryptic sites among alpha helices, and sometimes you even have an important conformational change. For instance, a change from alpha helix to beta sheet. These are more difficult to capture in our experience from the protein data bank, because often the electron density for one of the state is missing because of the flexibility. So yeah, so there are different classes, and this is also something that complicates the design of computational algorithms.
Okay, thank you very much. Zoe?
Yes, thank you very much, Francesco. Really great talk as always. Very nice what you can do with the tools. So I just want to ask, you mentioned it at some point, but which force field do you recommend for the identification of cryptic pockets with molecular dynamics?
So in our experience, both Charm 36M and Desamber work quite well, because they are both balanced for well-folded and also for partially unfolded states. Some of the older amber force fields are stabilizing too much the globular state. So they increase the barrier to the opening of the cavity.
I see. And what algorithm do you use to detect the pockets?
So we tried several, including F pockets. So we tried different approaches for me. So all the ones that I listed, we tried all of them.
Yeah, but I mean, which one did you find more successful?
On the student that is.
I see. Okay.
So F pocket is one that we use a lot.
Okay. And final aspect of my question: how do you discriminate then the true positives from the false positives? Because I guess when you run it, you identify not only the successful ones that you showed us, but you also have some smaller ones, or yeah.
So we had this suggestion in the original paper that we look at the cavities with the Swiss X, and we subtracted those that are found also with long simulation. But this is a bit arbitrary, because for some pockets, like the one for interleukin, you see it in both cases. So I'd say this is an open question.
Mm-hmm.
But I think that also the problem is that if you go to sufficiently high energy, you start unfolding and denaturing your proteins. So basically, you will start finding cryptic pockets everywhere.
Yeah.
Are they true or false? And then, in my opinion, it depends on how strongly your ligand will stabilize these cases. Because even if you have a partially unfolded state, there might be a potent ligand that is able to stabilize the state. So I think the question, it's difficult to address because it's a bit fuzzy.
Yeah.
It's not really well defined.
Do you calculate the free energy of the ligand binding then?
This is a thing that I think it's important to do. And we did in some cases, like with the COVID, where we had no answer to that. So then you can use method like metadynamics or one-opus to calculate the free energy of opening of the cavity. So if the penalty is too high, then clearly you have to discard it because it will be very difficult to design a ligand that is able to stabilize it.
Thank you very much for the wonderful webinar.
Thank you. So are there other
All right, do we have more questions from the audience? Okay, I don't see anyone raising their hand. Perhaps I can ask one last question, if no more questions from the participants. So you mentioned that when you're using the Swish algorithm for predicting the cryptic pockets, that the most successful cases are the ones where you use the Swish with water plus some probes. So how do you select the molecules to use?
This is a very good question. We have a project with Eleonora and with the University of Torino to try to find a good mix of mixed solvents. When we were working with, when Ben Cossing was still at UCB, they analyzed the clinically approved drugs to fragment them and to extract diverse set of fragments that would cover as much as possible the chemical space. But I think that is a good approach. And at the time we were using 100 different probes, but that's super expensive computationally. So you need to have a lot of GPUs to do that. So we are now looking at it, and I do not have a good answer because first of all, you have to define how you define the distance in chemical space. There are various matrices, of course. But then really the proof is in the pudding. So you need to have some probes that open the useful cavities and do not open the not useful one. And for that, I think you need experiments. So we are looking into this, but really I do not have an answer yet.
But generally in the research that is done in your group, you first go for the...
Well, we used a lot of benzene, but benzene is a very dirty fragment because it binds everywhere in the hydrophobic cavities. So I think heteroaromatic molecules are more useful because they are more similar to what you find in real drugs.
Thank you.
Okay. Thanks a lot. So thanks again for the invitation and thanks for following this webinar. And I hope to see you in person in the next meeting.
Thank you, Francesco.
Thank you.
Bye. Bye, everyone.